So thanks for the introduction. Hello everyone, my name is Xinyi Xu. I'm from Zhejiang University. Today I'm going to introduce you all of one of our neuroimaging genetic studies titled Unveiling Fetal Cortical Folding, Neuroimaging and Genetic Insights. So I have nothing to disclose. As we all know that the human fetal cerebral cortex undergoes a genetically orchestrated process during the prenatal period, which is a fundamental milestone of mammalian brain evolution. And any interference during this process may cause the long-term affected abnormalities. And with the development of advanced neuroimaging acquisition methods, and as shown here is the in-utero fetal brain MRI techniques, this process can be well observed, especially during the prenatal period. And the current consensus of cortical folding is that this process is under genetic, cellular, and biomechanical forces control, which collectively and interactively drive this process. And the integration of brain-wide gene expression atlas with the neuroimaging data has emerged as a promising method. And this method has been widely applied, especially among the adults, such as to study the genetic architecture of human brain and so on. And to our knowledge, there are two pioneering studies that correlated the MRI measurements with relevant genes using the ex-vivo and in-vivo fetal brains. However, they fell short in describing the cortical folding comprehensively. And as for the adult studies, they kind of fell short in providing the direct evidence of this process, especially during in the prenatal period. Therefore, in this study, we aim to use the advanced neuroimaging techniques, especially the in-utero fetal MRI technique and the public gene expression data to provide more direct insights of cortical folding. So specifically, first, we would like to understand the genetic basis of MRI-derived measurements of cortical folding. Secondly, we also want to investigate the relationship between morphological changes and the underlying microstructure alterations that are associated with cortical folding. In terms of the neuroimaging data, here we used our previously proposed two atlases. The first one is the spatiotemporal fetal brain T2-weighted atlas data, and the second one is the diffusion MRI fetal brain atlas data. So these two atlas can serve as the standard representation of the healthy fetal populations. Furthermore, we calculated the morphological and the microstructural characterization of cortical folding using the atlas data. Specifically, we computed the curvature as the morphological metric, which indicates the degree of cortical folding. And we also estimated the diffusion basis spectrum imaging model from the diffusion MRI data. So we usually call this model as DBSI for short. So basically, we could obtain four components from the DBSI model. The first one is fiber fraction, which was considered relating to the dendrites and axons, etc. And the second one is hindered fraction, which is thought to relate to the extracellular matrix. The third one is restricted fraction, and this may relate to the cellular component. So it represents the restricted diffusion within the cells, such as the neurons or the glial cells, etc. And the last one is water fraction, which accounts for the free diffusion of water molecules that are not significantly restricted by the cellular structures. And as for the genetic data, we use the pre-processed bulk tissue gene expression data, which were made publicly available as part of PsycEncode project. So in this project, each human brain specimen, there are like the regional dissection of up to 16 brain regions was performed, including the 11 cortical regions and 5 subcortical regions. So here we selectively incorporated the gene expression data from 4 prenatal specimens in order to match the gestational ages of our proposed fetal brain atlases. And to facilitate the comparisons between the developmental gene expression data and our MRI data, so we also created a set of cortical ROI labels corresponding to the anatomical dissection that used for the mRNA analysis. And then we can perform the correlation analysis to identify the genes that significantly correlated with the MRI-derived matrix of cortical folding. So for our results, we found 87 genes that are highly significantly correlated with the curvature at the early age. And to contextualize the genetic associations with biological processes, so we also performed the gene ontology enrichment analysis. And the 87 genes that correlated with curvature, we found that they were significantly enriched for like neuron genesis, neuron differentiation, and like the generation of neurons, and so on. As for the DBSI matrix, we found 79 genes that are significantly correlated with DBSI fiber fraction. And those genes were enriched for cell adhesion, biological adhesion, and circulatory system development, etc. In addition, we found 30 genes that correlated with hindered fraction. So these genes were enriched for cell adhesion, biological adhesion, and the extracellular matrix organization that we mentioned before. In addition, we also would like to examine the directional influence between the morphological changes of cortical folding as well as the underlying microstructure changes of cortical folding. So here we conducted a structural equation model to analyze this thing. And through this model, we found significant association between the early brain curvature and later DBSI fiber fraction and hinder fraction. So we think this result may indicate that the early degree of folding can influence the microstructure changes at a later gestational age. Finally, in summary, so here in our preliminary results, first we found some genes that significantly associated with the MRI-derived measurements, especially some microstructure measurements and morphological measurements of the cortical folding. And we also found some potential biological processes that may influence the cortical folding. And the last one that we also review some morphological and microstructure interactions during this important fetal brain development. So taken all, our study combines the advanced imaging modalities and genetic analysis to probe fetal cerebral cortex development and enhance our understanding of macro and microstructure interplay in the neurodevelopment. That's all I want to share today. Thank you so much for listening, and I'm happy to take any questions.
So people can come up to the microphone to ask questions, and there's also the app where you can ask questions online if you don't feel brave enough to come up to the microphone. We already have one online question, which is asking which tools were used to measure the gene enrichment and gene ontology analysis?
Sorry?
So which tools you use to do the gene ontology and gene enrichment analysis?
Oh, so it's a website called WebStat. Yeah, and I think it's the method for the enrichment analysis is over-representation analysis method.
Thank you so much.
Thank you.
If I may ask one more question, because I think we have time for about 2 or 3, unless someone else wants to jump up. I was curious whether you can speculate on any differences or overlap between the gene enrichment for the early and the late period, because you seem to find some difference in genes, but maybe there's also some overlap.
Yes, so there is some overlap between the two DBI symmetric related genes. That the two gene sets, they have some overlap, but from the microstructure related genes and to the morphological related genes, there was no overlap. Yeah, so.
Thank you. I think we're going to move on to the next speaker probably, because on a very tight schedule today.
Yeah. Thank you so much.